This is the first lecture for the second unit of the AP Biology course. So we've completed Unit 1, the Chemistry of Life, and now we're moving on to Unit 2, which is taking a look at cells, specifically their structure and function. And that is going to be the highlight of our first lecture. We'll take a look at cell structure, at the subcellular components. We'll look at the function of those components and also talk about why cells are very small. So our first lecture here is entitled Cell Structure and Function, and we'll take a look at the cell theory first, then a few comments on cell size. We'll learn the difference between prokaryotes and eukaryotes, and then take a closer look at cell structure and function. To begin, we'll discuss the cell theory. The cell theory states that, well, there's three postulates actually to the cell theory. First, that all organisms are composed of cells, and this was discovered and proposed by two scientists, a botanist, Matthias Schleiden, and a zoologist, Theodor Schwann. The second postulate to the cell theory says that cells come from pre-existing cells because they're self-reproducing. And German physician Rudolf Virchow was the discovering scientist for this. And third, cells are the basic units of structure and function in all organisms. Now, what about cell size? Why is it that cells are microscopic? Well, cells need a very large surface area of plasma membrane to do many functions: obtain necessary resources, eliminate waste products, acquire and dissipate thermal energy or heat, and exchange chemicals and energy with the environment. So you may be asking yourself, well, if they need a large surface area, then why aren't cells large? Well, here's why. There are limitations that restrict cell size and shape because smaller cells tend to need and have a higher surface area to volume ratio. As such, when a cell increases its volume, the relative surface area decreases, and therefore the demand for their internal resources increases. Now, I know that that's pretty hard to kind of grasp there, so let's go ahead and take a look at a visual example. Here we're going to be taking a look at three cubes. We're going to compare them here. So, and just imagine that these are cells. So here we have a 4 centimeter cube, meaning it's 4 centimeters on all four sides. Then we're going to split that 4 centimeter cube into 8 2 centimeter cubes. So that means each of those little cubes is 2 centimeters on a side. And finally over here on the right, we will take a look and divide into 64 1 centimeter cubes, so much smaller. Let's take a look at the total surface area of each cube. Okay, so if we take a look at the surface area of the cubes, we can see that the 1 4 centimeter cube has basically has an area of 96 centimeters. Whereas if we look at the 8 centimeter cubes, notice we have 192 square centimeters. And finally, the 64 1 centimeter cubes, the surface area is 384. Well, now let's compare the total volume. Notice that the volume ends up being the same in each case. So what we're demonstrating here is that when you go from a small cube, you're going to a large cube, you're decreasing the surface area and relatively increasing the volume. Well, what does this mean for surface area to volume? Well, take a look. That very large cube has a surface area to volume ratio of 1.5 to 1, which is pretty small. It's almost a 1-to-1 ratio. If we were to go ahead and increase that cube in its size more, we would see that ratio get closer to 1-to-1. That would mean that the given surface area of 1.5 here would need to absorb heat and chemicals and get rid of waste at a much higher rate because there's so much more volume. Notice the 8 2-centimeter cubes, the ratio is increased from 3 to 1. And finally, the smallest cubes, the surface area to volume ratio is 6 to 1, much higher. These little cubes or little cells will have a much easier time getting the oxygen and chemicals they need and also getting rid of the wastes they want to get rid of, because for their given volume of 1, they have a lot more surface area to make that exchange. So surface area to volume ratio is really important, and it is why cells have a small size. Now, surface area is really a very important concept in biology because cells and even organisms will even have specialized surfaces with which to increase their surface area and exchange molecules with the surrounding environment. So as we look at more complex cellular structures, and we're going to have a histology lab in here where we'll look at different cell shapes. You're going to notice that some cells really have a lot of folds in them, or perhaps long fibers that increase their surface area. And this is necessary to exchange materials with the environment. You have a lot of cells that take advantage of this. In particular, along the inside of your small intestine, there are cells called the microvilli cells. They have these lot of folds here. And so these epithelial cells that line your intestine have all these microvilli, these extensions that increase surface area because they want to increase the absorption of nutrients from the food that you eat. So more surface area means more exchange of chemicals with the environment. Now we're going to take a look at the difference between prokaryotes and eukaryotes. Prokaryotic cells are considered to be a lot more simplistic. They lack a nucleus. In fact, the word prokaryote, when it is directly translated from its original word stems, means before nut. And what this means is basically that these are the cells that appeared on Earth first. They're very primitive, and they appeared before a nucleus was found in cells. Now they say nut because the people that discovered the nucleus, the scientists that discovered it, thought it looked like a little nut inside of a cell. And so they called it a nut, but we now know it's a nucleus, right? So prokaryotic cells before nucleus, or they lack a nucleus, and they tend to be much smaller and have a more simple structure. They are not simple in their own rights. They have a lot of complexity to them, but we tend to say that in comparison to eukaryotic cells, they're much simpler. Now, eukaryotic cells, these have a true membrane-bound nucleus that houses or stores our DNA. The term eukaryotic cells means true nut, or in this case, true nucleus. These tend to be a lot more complex because there's a lot of what we call specialized membrane-bound organelles. So eukaryotic cells don't just have a nucleus. They have organelles that have a lot of membranes around them. Those are missing in bacteria cells or prokaryotic cells. They tend to be much larger and more complex than prokaryotes. And so now we're going to take a look at some structures that we find in cells, and we're going to focus mostly on eukaryotic cell structure. We'll touch on bacteria, cells, and other prokaryotes later on in the course. But for the purpose of understanding cell biology, we're going to focus on our eukaryotic cells, which of course would include human cells. Now, the first basic structure to look at in any cell, including a bacterial cell, is the cytosol. And this is basically the area just inside the plasma membrane. Actually, let me correct that. Not just inside, but it is inside the plasma membrane and outside of the organelles. So really, it includes the fluid portion of the cell known as the cytoplasm, as well as some interesting organelles called the ribosomes. Now, ribosomes you will also find in bacterial cells. So these are little tiny organelles that have a very important and conserved function. They serve to synthesize or make proteins, and they're found, as I've mentioned, in all forms of life, showing a common ancestry. This here is a transmission electron micrograph, basically a very highly magnified photo of the inside of a cell. And you can see the little dots that are there are the ribosomes. And later on, we'll talk about how and why they're attached to what we call the endoplasmic reticulum. Now, we mentioned that eukaryotes have a true nucleus. So this is the organelle in eukaryotic cells that contains the genetic material, or the DNA. This will be very significant when we look at the function of DNA and how proteins are made, because one of the interesting things is the code for making proteins is found on your DNA, which is stuck in the nucleus. As we mentioned, there's 6 feet of nucleus, or excuse me, 6 feet of DNA in each and every one of your nuclei. So it can't leave the nucleus, but yet it has the instructions to make proteins. And proteins are made on ribosomes and the endoplasmic reticulum outside of the nucleus. So how does that work? Well, we'll talk about that later when we look at protein synthesis in more detail. Now, the endomembrane system is a network of membranes that basically includes the nuclear envelope, which is the double membrane that surrounds the nucleus. And this is present in both plants and animals. So this is a plant cell, this is an animal cell. But it doesn't just include the nuclear envelope. It includes the endoplasmic reticulum, the Golgi apparatus, as well as some other membrane-bound organelles that we're going to talk about. Notice too, it also includes the plasma membrane, which is the membrane that encloses the outside of the cell. We have a whole special lecture that's going to go over the function of the plasma membrane and its importance, and also its detailed structure. Let's now look at working from the nucleus out. Let's kind of look at some of these interesting membrane-bound organelles that are unique to eukaryotes. We'll start with what's called the endoplasmic reticulum, which is sometimes referred to as the ER. Now, there are 2 forms of the endoplasmic reticulum, the rough and the smooth. So the rough endoplasmic reticulum is the part of the ER where there has the presence of ribosomes. It is studded, as it says there, with these little ribosomes. It works to compartmentalize the cell. Sorry for that interruption. I just didn't want to include the announcements on this video, but the endoplasmic reticulum works to compartmentalize the cell, to kind of section it off into different functioning compartments. And we'll talk more about that a little bit later when we look at compartmentalization. Now, the endoplasmic reticulum plays a key role in making or synthesizing proteins and also sorting them based on where they need to go, because your cells make a whole lot of proteins. Some of those proteins stay inside the cell to help the cell grow. Some are released from the cell and need to go to other parts of the body. And some might even be released as hormones or be synthesized and attached to other chemicals like lipids. So there's a lot going on with the proteins there. So as a result of that, the endoplasmic reticulum kind of plays the role of intracellular transport. It moves things around inside the cell. So it's kind of like a domestic shipping company, so to speak, that is going to move things. So the little ribosomes are like little factory workers that are making proteins, and the endoplasmic reticulum is like the assembly line, at least the rough ER. And the rough ER also works to ship and transport those proteins within the cell as far as where they need to go. Now, the smooth endoplasmic reticulum appears smooth because it lacks ribosomes. There's no ribosomes studding its surface. It has some different functions. Because there's no ribosomes, there's no proteins being made. So the functions of the smooth endoplasmic reticulum are a little bit different. It includes the detoxification of certain chemicals, as well as the synthesis or making of lipids. It also plays a key role in intracellular transport. Now, a membrane-bound organelle, as we work away from the nucleus again, notice the endoplasmic reticulum was kind of right outside the nucleus. Now we're moving a bit further away, and this blue organelle you see on the screen, it's not technically blue, they just kind of color-coded it so it looked different. It's called the Golgi apparatus. So it's another membrane-bound organelle, and it's not a connected membrane like the endoplasmic reticulum is, but it's more like a stack of flattened compartments. So almost like bubbles that have been flattened. It has some important functions, such as the correct folding and chemical modification of newly synthesized proteins, and also the packaging of proteins for protein trafficking. So you'll notice in the diagram below that the Golgi apparatus is making these little vesicles here that can attach to the plasma membrane and release the contents outside. This whole diagram here is showing you what we call the secretory pathway or secretory pathway, depending on how you want to pronounce it. Basically, this is how your cell will make chemicals that need to be secreted outside of the cell, such as neurotransmitters, that neurons that release neurotransmitters like norepinephrine and other things like dopamine inside your brain, but also the release of certain hormones and digestive enzymes from the cell. So you'll notice that those components, those chemicals, which are protein in nature, are made at the rough endoplasmic reticulum where the ribosomes are present. And so the proteins are made and then they are sent from the endoplasmic reticulum to the Golgi apparatus. The Golgi apparatus then goes ahead and modifies the proteins based on where they need to go and packages them into little vesicles. And if they need to go outside the cell, they will. And so they'll be released via the plasma membrane. We'll talk more about this excretion of chemicals outside of the plasma membrane a little bit later when we look at membrane transport. But notice that there's kind of like a cooperation between the endoplasmic reticulum and the Golgi apparatus here. So the Golgi body or Golgi apparatus is kind of like international shipping. It takes things outside of the country or the city of the cell and will take it to other areas and other cellular cities that need it. Now the lysosome is also a membrane-bound organelle. It is a cell that contains enzymes. Specifically, notice it's hydrolytic enzymes. And so hydrolytic enzymes do hydrolysis. And you may remember that hydrolysis is how we break large macromolecules down into smaller molecules. So this is why the lysosomes play an important role in intracellular digestion, in recycling organic materials, and even something known as the programmed cell death, because sometimes cells need to be done away with if they've reached the end of their life cycle. We have to get rid of them. Vacuoles are also membrane-bound sacs, but they have a different function. They're usually storage. They usually store and even release macromolecules and cellular waste products. And in plant cells, there's a very large central vacuole that its whole function is to store water. And that helps to maintain pressure inside plant cells. The peroxisome is a similar membrane-bound sac that catalyzes detoxifying reactions. So specifically, peroxisomes will work to get rid of toxins like hydrogen peroxide. And so they will basically break that toxin down so it can be removed from the cell. Now, one very important organelle is known as the mitochondrion. You may recall from previous science classes that the mitochondrion is called the powerhouse of the cell, or like the power company of the cell, because it's making most of the cell's energy. And of course, cellular energy comes in the form of adenosine triphosphate, or ATP. We'll talk about ATP in more detail later, but for right now, just understand that ATP is energy. It's the energy that all of our cells need to do life's work. Now, the mitochondrion is interesting because it has a double membrane system, with the outer membrane being smooth, making it almost look like a bean, but the inner membrane is highly convoluted and folded. And why do you think it's folded? Well, we talked about that earlier in this lecture, that the folding increases surface area, and also in this case provides compartments for many metabolic reactions to occur. So the folding of the membrane increases the surface area, which allows more ATP to be made by the mitochondria. It's a very important detail. And so we can see there are two interesting parts. So if we look at this, this is another scanning electron micrograph of actually looking inside the cell. And we can see the brown colored organelle here is the mitochondrion. Outside, the lighter colored area is the systole, or excuse me, the cytosol, which is like the cytoplasm and the cytoskeletal matrix and things like that. But if we focus on the mitochondria, the inside, the internal folds of the inner membrane are known as cristae. And the fluid inside the mitochondria, or mitochondrion, is known as the mitochondrial matrix. These two parts of the mitochondrion will be important when we look at what's called cellular respiration, which is how we go from the glucose and food that you eat to ATP. So really, the mitochondria is responsible for taking the byproducts of sugar and converting it into cellular energy. And we'll talk more about that later on in the course. Now, another very specialized organelle is the chloroplast, but you won't find this in animal cells. This is exclusive to photosynthetic cells, specifically plant cells and algae. Now, you'll notice, like the mitochondrion, it has a double outer membrane. Inside the chloroplast, there are some really cool structures that are important. Within the chloroplast, we have two very important things called the thylakoids and the stroma. Now, the thylakoids are like these green discs, or like green hockey pucks that are stacked on top of each other, almost like green coins. And the thylakoids, as we will learn later, are responsible for having the green pigment chlorophyll, which is important for photosynthesis. So a lot of photosynthesis and light absorption occurs where the thylakoids are. Now, the thylakoid, or the chloroplast, organizes these stacks of thylakoids into granum. They're like stacks of poker chips, one on top of each other. We call those the grana, or singular is granum. The stroma is the fluid inner portion of the chloroplast. It's outside the thylakoid, so it's outside the poker chips or the green hockey pucks, and is the fluid-filled portion. And we'll talk about the significance of that. So just like we saw that the inner mitochondrial membrane on the previous slide, the cristae, and the mitochondrial matrix are important for cellular respiration, we're going to come back and talk about why these thylakoids and stroma are very important for the process of photosynthesis, or how plants are able to take light energy and carbon dioxide and turn that into sugar. In this slide, we're going to go ahead and review everything, kind of put it together into four major sections of the cell. So we'll start with the nucleus. Of course, the nucleus is this portion over here. This is the location of the genome, which is basically the genetics of humans, or any organism, where all the DNA is. This is in the nucleus. We have this is where gene expression occurs and the regulation of certain genes, like turning them on and off. We'll talk about that again later. This is basically where DNA is organized into chromosomes, and it's also where ribosomes are made. So ribosomes, remember, those important little guys that make proteins, and we need a lot of them. So this is where you make a lot of ribosomes. Now the next section or quadrant of the cell is the cytosol. And this is the fluid-filled portion of the cell, all colored brown inside the cell membrane. And this helps to, this has some important functions too: coordination and response to the environment, coordination of metabolism, or all the chemical reactions that go on inside the cell. It makes what we call the proteome. We'll talk more about that later, not important now. And of course, it organizes and moves the cell via what we call cytoskeletal elements that you see here. And we may talk about those in a later lecture too. Now, the endomembrane system, as we talked about, includes the nuclear membrane, or the nuclear envelope that surrounds the nucleus. It also includes the endoplasmic reticulum, which includes both the rough and smooth ER. And we talked about how the endoplasmic reticulum is important for protein secretion and sorting. We'll talk about what glycalization is later. The smooth endoplasmic reticulum is important for lipid synthesis, and also the metabolic functions and accumulation of calcium. Now, the third is the Golgi apparatus. Remember the blue stacks of membranes, right? That also plays a role in protein secretion and sorting, and also glycalization. We'll talk more about that later. Don't worry about it right now. The lysosomes would be the little yellow bubbles there and the vacuoles, right? So they help to degrade organic molecules. They help to store them and also accumulate water, in particular in plant cells. The peroxisomes will break down toxic materials and also break down organic molecules via hydrolytic enzymes. And finally, 6 is the plasma membrane, which will be responsible for taking in and getting rid of certain ions and molecules. It will help with cell signaling, or how cells communicate with each other, and also how cells attach themselves to other cells and other objects. And fourth, the final section is what we call the semi-autonomous organelles, which include your mitochondria and chloroplasts in plants. They're called semi-autonomous because they actually reproduce themselves, as if acting almost like they're bacterial cells that kind of work by themselves. They even have their own DNA. So there's mitochondrial and chloroplast DNA. So that's interesting. We'll talk about that, the purpose and the origin of that later. Now, mitochondria, remember, they make ATP, powerhouse of the cell. They also can modify other organelles and produce heat, because that is an exothermic reaction. Chloroplasts, of course, are responsible for photosynthesis. The last and final slide here is going to be a quiz that I'm going to give you via the lecture. So you can see that I have some organelles labeled with just their functions and letters. And so in the quiz question that's about to pop up, or it might be a series of questions actually, you're going to identify the function of each of, or at least the organelle that each of the letters here is showing. And so we'll let you guys work on that over the next couple of minutes here.